Hello friends, myself Amit Belekar welcomes you to my channel Tips and Tricks by AMB. Today we are discussing about mechanism of action of penicillin. After going through this video, the learner will be able to describe mechanism of action of penicillin and describe bactericidal effect of penicillin. Penicillin is discovered by Alexander Fleming in 1929 in England. Benzyl penicillin has a narrow spectrum of activity. It is effective against gram-positive bacilli and cocci. Before dealing with mechanism of action, we have to discuss how bacterial cell wall synthesis can take place. Most of the bacteria consist of cell wall surrounding the cell membrane. In some bacteria, an additional layer is present in between cell wall and cell membrane. The gap between cell membrane and the cell wall is known as periplasmic space. Most gram-positive bacteria have periplasm where metabolic digestion occurs. Peptidoglycan is the most important component of the cell wall. It is a polymer made up of alternate bonding of N-acetyl-muramic acid and N-acetyl-glucosamine. These polymers are cross-linked with one another by chain of 4 amino acids. The bacterial cell wall maintains the shape and size of the organism and prevents the bacteria from bursting if fluid flows into it due to osmosis. Synthesis of bacterial cell wall depends upon the synthesis of peptidoglycan. Peptidoglycan synthesis occurs in cytoplasm and starts with addition of 5 amino acids to N-acetyl-muramic acid. Then N-acetyl-glucosamine is bonded with N-acetyl-muramic acid. This unit now acts as a precursor for peptidoglycan synthesis. This precursor unit is then transported through the cell membrane to the periplasm. These units incorporated in cell wall by forming polymer chains of these units. This process is called as transglycosylation. Then extensive cross-linking of these polymer chains takes place to form tough bacterial cell wall. This process is called as transpeptidation. For cross-linking of polymers, 2 enzymes are involved, viz. Transpeptidase and D-alanine carboxypeptidase. These enzymes, especially transpeptidase enzyme, are called as penicillin binding protein as penicillin can bind to them easily. These enzymes cross-links the polymers with the help of amino acid chain attached to N-acetyl-muramic acid moiety. Thus, several layers of peptidoglycan are developed to synthesize cell wall of bacteria. Gram-positive bacteria has several layers than gram-negative bacteria. Now we have to discuss about mechanism of action of penicillin. Penicillin have 2 rings in their structure, with a 4-membered ring that is also called as beta-lactam ring and a 5-membered ring which is also called as thiazolidine ring. Beta-lactam ring is having affinity to get bind with bacterial transpeptidase enzyme. Thus, it prevents cross-linking of peptidoglycan. Thus, it prevents transpeptidation process and inhibits cell wall synthesis of the bacteria. Bacteria have high internal osmotic pressure, and due to inhibition of cell wall synthesis, rigid cell wall does not form. Cell wall becomes weak. Because of outer lower osmotic pressure and internal high osmotic pressure and weak cell wall, bacterial cell burst. Thus, penicillin produces bactericidal effect. Now it is time to summarize. In this video, we had discussed about mechanism of action of penicillin and how it produces bactericidal effect in bacteria. Thank you for listening me patiently. If you have any queries, you can contact me on my mail address which is given below. If you want to watch more such videos, please like and subscribe my channel Tips and Tricks by AMB. Thank you once again.